Hello students, I am Akshay Deshpande and welcome to biology class. And today we will be discussing the last part of the chapter, that is the biomolecules. And in this particular session of the class, we are going to mainly concentrate on the enzymes. And we all know that enzymes are acting as the biocatalysts. Because in the last class, we have mainly discussed some of the important points about the metabolism. And if you remember that we have discussed one point regarding the enzymes. Okay, we had mentioned about the point that is the enzymes. Okay, and in this particular class, we will be trying to looking into about the enzymes. How do they actually work, and what are the some classification of enzymes, right? So let us start our discussion on the enzymes. So now, as you can see that enzymes are the proteins. Okay, so we can say that all of the enzymes that are occurring in the nature, okay, major of enzymes, we know that all of them are what? They are mainly proteins in a nature. They're mainly proteins. But however, there are some exceptions to this particular rule. And in the last class, we did mention about those exceptions, right? They are mainly the ribozymes. Okay, mainly the ribozymes. Okay, now ribozymes are basically the RNA molecules. Okay, they're mainly the RNA molecules. And these RNA molecules do have catalytic power. Okay, they mainly have catalytic power or catalytic ability, right? So similarly, even proteins also show this catalytic power. Okay, so what do you mean by catalytic power? That is forming, we can say, forming or breaking the bonds, mainly the breaking the bonds, forming and breaking the bonds. That is what we mean by the catalytic power, right? So ribozymes are the RNA molecules which show an exception to this particular rule, right? And an enzyme which can also be called as ribozyme is the ribosome. Remember, yes. So we know what is the function of the ribosome. Ribosome is mainly involved in the protein synthesis. It is mainly involved in the protein synthesis, right? Yes. So now protein synthesis, as I have told many times, that if it is a biopolymer of amino acids, and the amino acids are mainly joined together with the help of the peptide bond, right? So the formation of the peptide bond is mainly because of the catalytic ability of the ribosomes. Yes. So now, like any protein, as we have said that major of the enzymes are proteins, and we know that proteins are having different level of organization. Okay, and the level of organization, we are mainly referring it in the terms of the structure of proteins: primary structure, secondary structure, and tertiary structure, right? So similarly, we can see that as the enzymes are proteins, even they also exhibit the primary, secondary, and the tertiary structure. Okay, so if we are speaking of sequence, okay, if we are just referring to the sequence of amino acids, okay, if we are mainly speaking as the sequence of amino acids, then we can say that it is just the primary structure. Okay, it is the primary structure. However, if we are referring to the alpha helix, okay, if we are mainly referring to alpha helix and the beta pleated sheets, okay, a sheet-like arrangement of the bond or the amino acids, okay, then we are mainly referring it to the secondary structure. And if we are mainly referring to the formation of the disulfide bonds, okay, mainly the disulfide bonds, and also the folding of proteins, okay, folding of the peptide nature or protein, then it is called as what? Tertiary structure, right? So now we have discussed in our last class that the folding is involved in the associated with the tertiary structure, and ultimately the tertiary structure is important for one of the reason, and that reason is that it is giving the function. Okay, what kind of function? The biological, okay, mainly the biological function of proteins. Okay, it is the proteins. Okay, so the biological functioning of the proteins is determined by which level of organization or which structure. It is none other than the tertiary structure, right? So similarly, even in the case of enzymes, the activity, okay, the catalytic activity of the enzyme is determined by the tertiary structure. Okay, and at the same time, tertiary structure is also important for one of the another reason in the case of enzymes, that is the tertiary structure results in the formation of a pocket. Okay, pocket like structure mainly involves in the formation of pocket like structure, and this pocket like structure is called as the active site. It is called as what? The active site, right? So let us assume that if this is the enzyme, it is going to have a deep pocket like structure. Okay, so now what you are looking over here, this is called as the active site. Okay, and what is the function of the active site? Active site is the site where the substrate binds to the enzyme. So it is mainly the region present in the enzyme where the substrate can bind into, and that part is referred as the active site. And active site is mainly having the catalytic power. It is having the catalytic power. Okay, so now we can say that the part or the region of the enzyme that is having the catalytic ability or catalytic power is none other than the active site. So we can say that not all or whole of the enzyme is actually having the catalytic ability. Okay, so only the active site is having this power, that is the catalytic power. So here we have mentioned that the active site is formed by the folding of proteins, and the active site is actually a pocket or a crevice, okay, where the substrate actually binds. So through this particular site, you can see that the catalysis actually occurs. Enzymes enzymatic catalysts and the inorganic catalysts are quite different in their properties. There is one important property that differentiates the enzymatic catalysts and inorganic catalysts, right? That is the temperature specificity. We can say that in the case of inorganic catalysts, in the case of the inorganic catalysts, they can work at high temperatures. They can easily work at high temperatures. But in the case of organic catalysts, okay, organic catalysts, mainly we are speaking of enzymes. And we know that enzymes means mainly the proteins. Now these enzymes, that is the proteinaceous enzymes, they work at a narrow range of temperature. They don't work at every temperature. They work at a very narrow range of temperature. So we can say that at high temperature, what happens is that enzyme activity is degraded or enzyme is destroyed. Enzyme is destroyed. So this is one drawback of the protein enzymes, that they work at a very specific temperature, and beyond that particular temperature, what happens is that the activity of the enzyme is lost. right? But however, there are some exceptions to this particular rule. There are some enzymes, proteinaceous enzyme itself, okay, and they are thermostable. They are mainly thermostable. That is, they are mainly stable at the high temperatures. So these thermostable enzymes are mainly derived from the thermophilic organisms. Thermophilic organisms. Now, thermophilic organisms are some organisms which mainly live at or live in the high temperature region, such as the thermal vents, mainly at the thermal vents, or we can even say that the hot water springs, hot water springs, right? Where the temperature is around 80 to 90 degrees Celsius. So even at such a high temperature, you can see that these enzymes, in the case of the thermophilic organisms, they are functional and they retain the catalytic property or catalytic ability, right? But however, not all enzymes do have this property, right? Major of these proteinaceous enzyme work at a very narrow range of temperature, and if the temperature increases more than the working area or working region, you can see that the enzymes become inactive. Now, moving on, chemical reactions, we have to. Now, let us look into some of the changes, that is mainly the chemical reactions, right? So, the changes can be either physical or they can be either chemical. So now, in the case of physical changes, you see that there is no change in the bonds, but however, there is a change in matter, right? We are having an example that is melting of ice, melting of ice to water. Melting of ice to water, we can consider it as a physical change. But in the case of the chemical changes, there will be change in the bonds. Either the bonds are broken or either the bonds are created. Now here we are having an example for this, that is a barium hydroxide with reacts with the H2SO4, that is sulfuric acid, to give us a barium sulfate and the water. Now we can see that we are specifying this reaction in the terms of rate. We can tell the reaction occurring in the terms of rate, that is the rate of change of a formation of a product, right? So here we are having delta P and a delta T. So we can say that the rate of change or the chemical process can be referred in the amount of product formed per unit time. So this is the equation that tells us about the rate of formation of the product in a unit time. So now the rate can also be called as velocity if we are considering the direction. Okay, if we are considering the direction, in which particular direction the reaction is occurring. So how fast the rate of reaction is occurring, then we can say that it is called as the velocity. So now, one of the important aspect, as we mentioned just a moment ago, that the whole chemical reactions are affected by the temperature. Temperature and pressure plays a very important role in the rate of reactions occurring. Either the reactions can occur at a slow or can occur fast. But however, we are having a general thumb rule for this. And we have mentioned it over here that the rate of reaction can double with the every 10-degree increase in the temperature. Okay, so if I'm going to increase the temperature around 10 degrees Celsius, the reaction rate, okay, the reaction rate increases. The reaction rate increases. So if I decrease the temperature by around 10 degrees Celsius, the reaction rate halves. Okay, reduces by half. We can say it is reduces by half. This is what we mean by the general thumb rule regarding the process or the rate at which the reactions actually go. So now here we are having certain catalyzed reactions, and generally the catalyzed reactions are more faster than the uncatalyzed reactions. And here we are actually having an example, that is the carbon dioxide when it fuses with the water. Okay, you can see that it is converted into carbonic acid, and the enzyme that is involved in this particular reaction is called as the carbonic anhydrase. You will study about this reaction in the respiration chapter. Okay, so now if there is no carbonic anhydrase, when there is no carbonic anhydrase, okay, so when there is no carbonic anhydrase, you can see that the rate of formation of the carbonic acid is around 200 molecules per hour. Okay, yes, so you can see that the formation of carbonic acid is very less when there is no carbonic anhydrase. But however, if we start to add the carbonic anhydrase, you can see that the formation of the carbonic acid is increased. So how much the molecule will increase? You can see that the formation of carbonic acid is going to be around 600,000 molecules per second, right? So you can see in the absence of carbonic anhydrase, the number of carbonic acid molecules were formed around 200 per hour. But now you can see that the moment we start to add the carbonic anhydrase, the formation of the carbonic acid has increased from 200 to the 600 molecules per second. So we can say that around 10 million times the reaction has sped up. Okay, 10 million times, okay, around 10 million times the reaction has speeded up. That is what we can derive. So hence we can say that ultimately by adding of the enzymes or catalyst, the reactions can speed up. This is what the reactions tell us. Now, similarly, many reactions are either single-step reactions or they are multi-step reactions. And generally, in the case of multi-step reactions, they are catalyzed by different type of enzymes. Okay, you do see certain multi-step reactions where a single kind of enzyme is involved in all the steps, but general of the case, most of the cases, you see that all of these steps in a multi-step metabolic pathway or multi-step enzyme catalyzed reaction, at each step a different enzyme is actually involved in the process. And we are having an example for this, that is the glucose, which is converted into pyruvic acid. So if I have to ask you the question, what is the number of step that is involved in breaking down of glucose into pyruvic acid? And this reaction, what we are looking over here, is actually called as the glycolysis, if I am not wrong, right? It is called as the glycolysis. We have discussed about the term called as the glycogenolysis, glycogenesis, gluconeogenesis, and also the glycolysis. So in the case of glycolysis, this step, that is the glucose to pyruvic acid, is called as the glycolysis, and the number of step that is involved in the glycolysis is around a 10-step reaction. So we know that it is a 10-step reaction, and each of these steps is catalyzed by a different enzyme. Not all the 10 steps are catalyzed by a single enzyme, right? All of these 10 steps are catalyzed by different enzyme itself. We will be discussing about the enzymes that catalyze the glycolytic reactions in the respiration chapter. Now, let us also look into how the enzyme actually work. Okay, let us look into the mechanism of enzyme actions. So here we are having a very simplest form of equation, that is S to P. The S here mainly stands for the substrate. It is mainly the substrate, and the P mainly stands for the product. Mainly stands for the product, right? But however, the equation over here looks to be very simple, but however it isn't. You can see that the substrate has to bind with the enzyme, and this enzyme will along with the substrate is resulting in the formation of a complex called as the enzyme substrate complex, and this enzyme substrate complex will result in the formation of enzyme plus product. This is the reaction that we all have studied, right? So now this is called as the transient state or transition molecule. transient compound. It is called as a transient compound. Why it is called as a transient compound? Transient mainly means a temporary, right? So it means that it is not a permanently formed substance. It is just a temporary association of substrate with the enzyme, and the enzyme can do the necessary changes in the substrate molecule and result in the formation of the product. And that is what we are interested in. Here, we are actually having a graph that tells about the working mechanism of the enzymes. So now you can look at the graph that this dotted line mainly indicates the substrate, and this is the x-axis, which mainly tells about the progress of the reaction. And here we are having is the potential energy. And there are two curves that you can see over here, right? So one of the curve mainly tells about the presence of enzyme and with respect to the activation energy, and one more curve, the bigger curve, tells mainly about the in the absence of enzyme and the activation energy. So now let us look into the concept of what is activation energy. Activation energy. Now activation energy is the energy difference. Okay, it is mainly the energy difference, right? Now the energy difference that is lying between the substrate and the transition state. Okay, now what do you mean by transition state? Transition state is a temporary state. Okay, it is the state at which the enzyme substrate complex exists. Okay, this complex, that is the enzyme and substrate, both of them are together, and that is called as the enzyme substrate complex. Okay, so the moment or the duration of this enzyme substrate complex, it is a temporary. It is not completely observed for a long moment of time. Okay, and to achieve this particular state, you can see that they have given the arrow mark over here. Okay, so you can see that from substrate to the transition state, it is a state at which the enzyme substrate complex exists. It is present. Okay, so you can see that to achieve the transition state, the energy is required. Okay, and this difference between the energy is called as the activation energy. Okay, so now in the absence of the enzyme, the energy difference is very high. Hence we can say that the time to reach the, what we can say, transition state is high. Okay, in the absence of enzyme, the time to reach the transition state is more. Okay, yes. So now, however, you can see that this activation energy or the energy difference in the case of presence of enzyme is decreased. That is why the arrow mark is very less over here. Okay, so you can see that enzymes mainly work by bringing down this energy difference between the substrate and the transition state. That is how we can see that enzymes actually work, right? So in the absence of enzyme, the time to reach this what we can say state is more. Okay, but however, we can even observe that in the presence of enzyme, the energy difference between the substrate and the transition state is decreased. It is lowered. Okay, and this lowering of the energy difference is mainly because of the what we can say, the enzyme. It is mainly due to the presence of the enzyme. So hence, in a nutshell, we can conclude that enzymes, okay, this statement is very important to remember. Enzymes mainly work by decreasing the activation energy. Okay, or we can even say lowering the activation energy. They bring down that activation energy. Okay, so enzymes mainly work by lowering or decreasing or bringing down of the activation energy. And we know what is activation energy. It is the energy difference that is existing between the substrate and the transition state. So we also know what is transition state. Transition state is a state at which the enzyme substrate complex is existing. Enzyme substrate complex is existing. That is called as the transition state. Okay, so generally what happens is that the energy difference is more when there is no enzyme, but however, as soon as you start to add the enzyme, the energy difference to achieve or to reach that state, that is the transition state, is decreased. And how it is decreased? It is mainly by lowering the activation energy. Okay, so we can say that in a less moment of time, in a very short period of time, the enzyme substrate complex is formed, and as a result you can see that later the product is actually formed over here. So now here you can see that the product or the product that is formed is lesser than the substrate. Okay, it is below than the substrate, and such kind of reaction is called as the exothermic reaction. Okay, so if you see that the product is lesser or lower than the substrate in the graph, we can say that this kind of reactions are called as the exothermic reactions. So now we can look into the nature of the enzyme actions. Okay, so exactly the same what we can say, the statement we had written earlier, that is a enzyme plus substrate, we will get an enzyme substrate complex. Later the enzyme will do all the necessary change in the bond of the substrate. As a result, there is a formation of the enzyme product complex, and finally you can see that enzyme and product are formed. Now enzyme is not consumed in the reaction. Remember, the enzyme is never taking part in the reaction itself. It is never consumed in the reaction. It is released, and finally this enzyme is free to catalyze another reaction, and finally we get the product later. Okay, so you can see that what are the steps of the enzyme reactions or actions. So the first step that is involved in the enzyme action is that the substrate mainly binds to the active site of the enzyme. Okay, an active site is the site where the substrate can fit into that particular enzyme. Remember, the active site is flexible. Also, the active site is flexible. Okay, it is actually flexible in nature. There is no rigidity. Which means that even if a substrate with different shape and structure, it can still bind to the active site of the enzyme. Okay, there is no stringent rule, or there is no hard and fast rule that the shape of the the shape of the active site of the enzyme should be complementary with the shape and the structure of the substrate. Okay, once upon a time we used to think it in that way, but however now it is clear that there is no such stringent requirement. Okay, the active site can change its shape and structure according to the shape of the substrate. Hence we say that active site is actually flexible in a nature. Next, the second step is enzyme alters its shape to bind more tightly to the substrate. So this second point itself will tell us that active site is mainly flexible in nature. After the substrate binds to the active site can slightly modify itself according to the shape of the active, we can say that is the substrate, and it is now ready to catalyze the reaction. So you can see that the third point is the substrate bonds are broken or they are rearranged, and finally the enzyme product complex is actually formed. This step over here. The last step we are having is the product is released and the enzyme is ready for the another set of reaction. Okay, product is also released, enzyme is also now free, and this enzyme is now ready for catalyzing the next set of reactions. So this is what we can say the nature of enzyme action. Now let us look into some of the important factors that affect the enzyme activity, right? So here let us mention about three important factors. The first we are having is the pH. And the second factor we are having is the temperature, which we have mentioned earlier. Okay, temperature is also an important factor. And finally, the substrate concentration. Okay, substrate concentration. Now, these graphs are already mentioned in your textbooks. Okay, so you have to remember these graphs properly. Okay, because they may ask you which of the following graph is correct or not correct. Okay, or they may ask you graphical based questions over here, that which of the following graphs tell about the activity of the enzyme in the high pH or low pH or something like that. Okay, so there's every possibility, every chance that they may ask you about the graphical based questions, right? Hence you have to remember these graphs more appropriately. So now let us consider the first, that is the pH. So now we know what is a pH. That is, it mainly tells about the acidity and alkaline nature. Right? Acidity means presence of more H plus ions. Alkalinity mainly means the presence of more OH ions. Okay, yeah, minus. Sorry, this minus. Now we can say that in high pH conditions or in high alkaline conditions, the enzyme becomes inactive. Enzyme becomes inactive. Right? Now, this statement, we can say that it is more evident over here. So here the x-axis, we can see that the pH is low. Okay, we can write this as the low pH. Low pH mainly means acidic pH condition, acidic conditions. This is, we can say, it is a high pH. Okay, so you can see that when the pH is completely low, the enzyme activity is completely nil. But as the pH goes on increasing, you can see that the activity also increases, but only up to one certain particular extent. Okay, now this, right? So now if you draw a line, the peak of that particular graph to the x-axis, you can see that the best activity that you can find of the enzyme is a kind of neutral. Right? Don't you think this graph is exactly in between? Right? The peak that we have seen is in between the high and low pH. Okay, so you can see that the best activity can be ranging from around the 6.8 to around the 7.2. Okay, so this is the pH range. You can see that very good activity is observed of the enzyme. But now, however, if the pH, if it goes on increasing, it becomes high pH, that is more basic in nature, again the curve goes on decreasing, and finally at highest pH level, the activity is completely nil. Okay, because the enzyme is destroyed, or we can say that a catalytic property or catalytic ability of the enzyme is completely lost. Now, similarly, same kind of graph is also observed in the case of temperature too, right? So you can see this is, we can say, very low temperature, and at this point we can say it as the high temperature. So at the lowest temperature, you can see that enzymes are completely inactive. They are not working. As the temperature goes on increasing, even the activity of the enzyme increases, and at one peak, if you draw the line from the peak, you can see that the functioning of the enzyme is exactly in between the low and the high. Okay, hence we can say at a moderate temperature, okay, at the moderate temperature, you can see that the enzymes work exactly perfect. Okay, so what is the temperature range? You can say generally we say that it is around 25 degrees Celsius to around 30 degrees Celsius. This is a very narrow specific range. Okay, hence in the previous, just a moment of a previous slide, we mentioned that the enzymes work at a very narrow range of temperature. Okay, but inorganic catalysts, they can work even at high range of temperatures. Okay, they are not temperature dependent. Even at high temperatures, they can work happily. Right, that is the inorganic catalysts. But the graph itself indicates that the enzymes, proteinaceous enzyme, mainly require a very narrow range of temperature, moderate range of temperature. And this moderate range of temperature, as you can see, it is around 25 degrees Celsius to 30 degrees Celsius, right? Now, as if we go on increasing the temperature furthermore, you can see that the activity of the enzyme goes on decreasing, and finally there is no activity at all, right? Because the enzyme is completely destroyed. You can see that at higher temperature, the activity of the enzyme is completely destroyed. So these two graphs mainly, that is A graph and B graph, mainly depict the pH and the temperature as a role playing in the activity of the enzyme. Now finally, let us look into the last graph, that is the substrate concentration. Okay, how do the substrate concentration affect the activity of the enzyme? So now what we can do is that we can take a small beaker, okay, and let us assume that in this beaker we are having an enzyme solution, okay, and let us assume that there are 10 enzyme molecules, okay, there are around 10 enzyme molecules, right? Yes. Now what I can do is I will start to add the substrate, right? Yes. So when there is no substrate in the solution, all the 10 enzyme molecules are free. They are ready to do the catalysis. So now as I start adding the substrate molecule, for example, if I add one molecule, what happens? The reaction slowly increases. Okay, now I will add one more molecule. Slightly the reaction increases, right? I will add one more molecule and slow on. So you can see that as I go on adding the substrate, the reaction increases, right? Now, but however, you can see that the reaction rate or the enzyme catalysis increases only up to a certain extent, beyond which it becomes completely stagnant or it becomes asymptote. Okay, now this what you are looking over here is called as the asymptote. Yes. Now what do you mean by asymptote? Now this line, the dotted line and the curved line goes, they move parallelly. Okay, they never cross each other. The graph will not cross the line, the curved line will not cross the dotted line over here, right? Yes. So now why did the reaction go stagnant? Why it became stagnant? Because if I go on adding the substrate molecules, what happens is that more and more number of enzyme molecules are being engaged. Okay, but we know that there are only 10 enzyme molecules. If I add 11th substrate molecule, okay, if I add 11th substrate molecule, do you think there will be any reaction? No, there will be no reaction because there is no 11th molecule present in the reaction itself, right? So it is this particular moment, at this point when I add the 11th substrate molecule, the reaction hits the highest. Okay, at this particular moment you can see that there is no further increase in the rate of reaction, right? So now we can see that the Vmax, we call it as the velocity of the reaction, maximum velocity of the reaction, we call it as the y-axis. Okay, and S mainly stands for the substrate concentration. Okay, mainly the substrate concentration. Okay, so you can see as the substrate concentration is increasing, even the reaction or the velocity of the reaction is also increasing. Okay, but only up to a certain extent. Okay, because if the substrate concentration increases more than that of the enzyme concentration, you can see that all of the enzyme molecules are saturated, no free enzyme molecules are present, and hence the reaction hits asymptote or becomes completely stagnant. Okay, so now let us look into this, that is called as the Vmax by 2. Okay, exactly half velocity of the whole reaction, we call it as the Vmax, half of velocity of the reaction, hence we call it as Vmax by 2. And if I try to put a dotted line and extrapolate it to the x-axis, we will get the Km. It is called as the Michaelis constant. Okay, now don't worry what is Michaelis constant. It is mainly an equation called as Michaelis-Menten reaction. Okay, these were the two scientists who actually described the enzyme kinetics. Okay, and it is completely mathematical. What we can say, it's completely physical chemistry. Okay, so I don't think it is required for our discussion over here. But one thing is very absolute, that is the rate of reaction increases only up to a certain extent, beyond which there will be no increase. Okay, even if the substrate concentration increases beyond the enzyme concentration, there is no increase in the reaction. Okay, rate of reaction or velocity of the reaction. This is the ultimate takeaway from this point what we have discussed. So now let us look into the inhibition of the enzymes, right? So now, can we inhibit the enzyme activity? Exactly, we can do that. Okay, and we are having special kind of molecules called as the inhibitors. Okay, now what do the inhibitors do? Now inhibitors, okay, inhibitors, they are very similar to the substrate molecules. Okay, they are very similar to substrate molecules. In what terms they are similar? They are mainly similar in the terms of the structure. Okay, in the terms of a structure, they are very similar over here. Okay, so now as they are similar to the substrate molecules, you can see that if I add inhibitor instead of the substrate, the inhibitor can bind to the enzyme. They can bind to enzyme. Okay, but however, after binding to enzyme, there will be no catalysis. Okay, there will be no catalysis, right? So there will be no catalysis in the sense the enzyme is actually inactivated. So inhibitors are important because they can actually inhibit the enzyme activity, right? So now we are having one special kind of mechanism through which we can inhibit the enzyme activity. It is called as the competitive inhibitors or competitive inhibition mechanism. So now what happens is that in a reaction, okay, in an enzymatic reaction, this is the enzyme solution. I will add both, okay, the inhibitor and also the substrate. Okay, this is the enzyme solution. Okay, this is the enzyme solution. I will add both the substrate and also the inhibitor. So what happens is that the inhibitor molecules will compete with the substrate for the active site of the enzyme. Okay, hence it is called as the competitive inhibition. Okay, but however, this inhibition is not permanent. Okay, not permanent. Okay, it is temporary. It is temporary. Remember this. Okay, it is temporary. Okay, now how can we reverse this? If I increase the substrate concentration, okay, what happens is that the inhibition can be reversed. Remember this. So competitive inhibition mechanism is mainly used to control the bacterial pathogen. Okay, so the inhibition mechanism application that you see is mainly for the controlling the bacterial pathogens. So now let us look into the classification and nomenclature of the enzymes. Remember that we are actually having around 6 different classes of enzymes. Okay, so if you ask how many enzymes are present, around 6 classes are actually present, and each class is having around 4 to 13 different subclasses. Okay, around 4 to 13 different subclasses which are having its own 4-digit number. Okay, it is having around 4-digit number that we give for each enzyme over here. Okay, it is a very elaborate, and the thing is that the nomenclature and the classification of enzymes changes every, what we can say, few years or so, because new enzymes are described and new enzymes are added to the classification list. Okay, but let us look into the 6 general classification that we know. The first one we are having is the oxide reductases, or also called as the dehydrogenases. Now they are mainly involved in what we can say oxidation or reduction kind of reactions between the two substrate. Here you can see that the initial part of the reaction, S is actually reduced, substrate is reduced, and what we can say, the S prime or S dash is actually oxidized. Now, in the presence of this kind of enzymes, you can see that the S becomes oxidized and S prime or S dash becomes the reduced finally at the end. Okay. For example, we are having alcohol dehydrogenase. Okay, it is having the alcohol dehydrogenase. Now it is an example for a dehydrogenase. Okay, similarly, oxidation reductases, there are many enzymes for us. The next one we are having is the transferase. As the name itself indicates, it is mainly involved in the transferring of the groups. Okay, for example, the substrate is having this group molecule, it should be transferred to this S prime, right? That is what this enzyme is doing. The next category we are having is the hydrolases. Okay, now hydrolases of the ether, peptide, and glycosidic bonds, carbon-carbon bonds, carbon halide bonds, and even phosphorus-nitrogen bonds. Okay, and generally water is involved in this kind of reaction. The next one we are having is the lyases. Even in the case of lyases, there will be removal of certain groups, but however, the mechanism is other than the hydrolysis. Here, water is not formed over here. Water element is not used. Okay, and the product will be having a double bond. Right? You can see that X and Y molecules are none other than some groups which we are requiring to remove it. So at the end of the reaction, in the presence of the lyases enzyme, you can see that X and Y are separated from the C and this group, and you can see that there's a double bond formed between these two carbon molecules. The next one we are having is the isomerases, which are mainly involved in interconversion of the optical, geometrical, positional isomers. Okay, just converting from, for example, if there's a carbon, this is O and this is H. What we can see, the enzymes can do in this way. The H can be changed its position from here to here. So the O comes from left side to the right side. This is called as isomerases. These enzymes, okay, this kind of conversion are called as the optical conversion, interconversion. The last one we are having is the ligases. Ligases joining, okay, linking of two compounds, okay, that is the carbon oxygen, okay, carbon sulfur, and carbon nitrogen, phosphorus oxygen, right? This is what these enzymes mainly do. So now there are other important factors called as the cofactors that they are required for the activity of the proteins. Okay, mainly the enzymes, what we can say. The cofactors are generally non-proteinaceous constituents. Okay, enzyme, as we said, that they are mainly made up of proteins, but there is no rule that they should be completely made up of proteins. They also contain some non-protein constituents, which we refer them as cofactors. Okay, now we are having a term called as the apoenzyme, which is mainly called as the incomplete enzyme or non-functional enzyme. Okay, so if I add the cofactor, plus the apoenzyme, okay, if we add both these, we get a functional enzyme. Okay, we get functional enzyme. Okay, and the functional enzyme is called as the holo enzyme. It is called as the holo enzyme. Remember, apoenzyme is none other than the non-functional enzymes. So we are actually having three types of cofactors. The first is called as the prosthetic groups, coenzymes, and metal ions. The prosthetic groups mainly consider the organic compounds. For example, in the case of catalase and peroxidase, these two are enzymes. Okay, they are mainly involved in degradation of hydrogen peroxide, H2O2. Okay, so both of these enzymes are having heme group as the prosthetic groups. Coenzymes, we can see that they mainly bind temporarily, okay, with the enzymes, and interestingly, many of them are vitamins. For example, the NAD stands for nicotinamide dinucleotide, okay, and NADP means nicotinamide dinucleotide phosphate. Okay, it is mainly having vitamin B3, that is the niacin. And in some cases, you also find the metal ions. For example, in the case of carboxypeptidase, now this is actually a protease, remember. Okay, now what is a protease? Protease is actually involved in breaking down the proteins, right? Now carboxypeptidase is an enzyme which is mainly involved in breaking down of the proteins, and it is requiring a metal ion called as the zinc. Right, so we have discussed some of the important points regarding the enzymes. Now what we can do is we can start discussing few questions on this particular part. Now, the first question we are having is enzymes are proteins and their tertiary structure involves folds, and this results in the formation of dash that helps in catalysis. Okay, the results in the formation of active site, allosteric site, catalysis, disulfide bond. It is none other than the active site. Okay, but however, we are also finding one more site called as the allosteric site, which controls the enzyme activity. Okay, now allosteric site is not involved, in catalysis. Remember this. It is not involved in catalysis. Remember this. Okay. Yes. So yeah, C is not the answer. D is also not the answer. Okay. It is the catalyst active site that is formed due to the folding and of the enzymes. Which of the following is true about the inorganic catalysts? They are similar to enzymes in terms of structure. They have active site. They can work at high temperatures. They are used up in the reaction. Okay. So now, inorganic catalysts. What is true about the inorganic catalysts, right? They are similar to enzymes in terms of structure. No. They have active site. No. They can work at high temperatures. Exactly. C is our right answer. They can work at high temperatures. Okay. But unlike in the case of enzymes, they work at a very restricted, narrow range of temperatures only. The next questions. The cavity or pocket-like structure in the enzymes play role in catalysis, speed of the reaction, regulation of substrate concentration, none of the above. They play a role in the catalysis, that is breaking or forming of the bonds. The next one, which of the following statement is about the enzyme is correct? Okay, they are temperature specific. Some enzymes work at high temperatures. They are not consumed in the reaction. All of the above. Exactly, all of the above mentioned statements are actually true. They are temperature specific, but some enzymes can also work at high temperatures, and they are mainly derived from the thermophilic organism. Thermophilic organisms, right? We have mentioned about this. Now, so hence we can say that all of the above is the answer. Which of the following molecules have catalytic activity? RNA and DNA, RNA and protein, protein and DNA, proteins only. We know some RNA molecules called as the ribozymes do have the catalytic activity, hence RNA and protein. Okay, DNA does not have any kind of catalytic activity. Okay, and proteins alone are not having the catalytic activity. Even RNA molecules do have certain degree of catalytic activity. The thermostable enzymes are derived from organisms that live in the oceans, organisms that live in hot sulfur springs, organisms that live in high altitude. All of the above. We know they mainly live in the thermal vents and hot sulfur springs. That is also called as the hot water springs. And these hot water springs mainly contain high amount of sulfur. Hence, they are also referred as hot sulfur springs. The next one, which of the following statement is correct? Physical change involves a breaking of bond that change the shape. Chemical change involves a breaking of bond. Major of enzymes are not damaged at high temperatures. Ice melting to water is an example for chemical change, right? Melting of ice to water is actually a physical change. And major of the enzymes are actually damaged at high temperatures, right? And chemical change mainly involves the breaking of bonds. Exactly, that is a correct statement. And physical change involves breaking of bond that change the shape. No, it involves a change in the state. Okay, from one state to another state, from one matter to another matter, but there is no involved in changing the or breaking the bonds. Yes. The next one, which of the following statement is true? A rate can be constant if we mention the direction. Rate can be direction if we mention the velocity. Okay, the rate doubles with a 10 degrees decrease in the temperature. Rate reduces to half with the 10 degrees decrease in the temperature. Exactly, the rate of the reaction decreases with the decreases to half with the 10 degrees decrease in the temperature. Okay, but the rate doubles with the 10 degrees increase, not the decrease. The rate of the rate can be velocity. This is not direction or the constant. Okay, rate can be velocity if we consider the direction. Okay, so the first two statements are also not true over here. Okay, along with the third statement. So the D statement is actually the true statement. The next one, which of the following enzyme is involved in the catalysis? Carbon dioxide to carbonic acid. Okay, peroxidase, catalase, carbonic anhydrase, hexokinase. We know it is mainly the carbonic anhydrase that is involved in this particular reaction. So C is our right answer. The next question, which of the following statement is incorrect? 600,000 carbonic acid molecules are formed in the absence of carbonic anhydrase. Okay, and the addition of carbonic anhydrase speeds up the reaction about 10 million times. Okay, a multi-step reaction are catalyzed by same or multi set of enzymes or different set of enzymes. The rate of enzyme catalyzed reactions are higher than that of the uncatalyzed reactions. They have asked mainly about the incorrect statement. Okay, and you can see that 600,000 carbonic acid molecules are formed in the presence, not in the absence. We can say A is our right answer and the incorrect statement. Remaining all are actually the true statements over here. Moving on, which of the following is a metabolic active pathway? Okay, glycolysis, Krebs cycle, gluconeogenesis, all of the above. We can say all of the above mentioned are actually the metabolic pathways. The next one, which of the following is not true about enzymes? Catalysis, altering the rate of reaction, cannot be inhibited, working at a specific temperature. C is not true. They cannot be inhibited. Actually, they can be inhibited, right? So the C statement is actually not true or incorrect. Which of the following is formed during the transition state? Enzyme product complex, enzyme substrate complex, enzyme molecular complex, and none of the above. We know it is the temporary association of the enzyme and substrate is called as the enzyme substrate complex, and the moment at which it forms is called as the transition state. The next one we are having is the difference between the substrate and the transition state is the turnover, enzyme substrate complex, the enzyme product complex, activation energy. Okay, the difference between these two is actually the activation energy. D is the right answer. The next one, which of the following statement is not true? The enzyme is used in the reaction. The product is released from the enzyme. Some RNA molecules have catalytic properties. Yes to P is the simplest equation for enzyme reaction. So you can see this statement is true. This is also true. This is also true. But not true is A. That is, enzyme is used in the reaction. Enzymes are never used in the reaction. They will be ready at the end of the reaction to catalyze another set of reactions. The next one, which of the following graph shows the activation energy properly in the presence of enzyme? Yes, as I had said, you're going to get a certain graphical based questions. Okay, so the x-axis and y-axis, P mainly stands for the potential energy. Okay, and you can see that C and D options are very much near to our what we can say graph what we had seen, right? This was the graph. Okay, and this is in the presence of enzyme and this is in the absence of enzyme, right? So D is mainly this graph and C is mainly this graph. In the presence of enzyme is the C part. Okay, the smaller graph is smaller curve is actually the presence of enzyme, and you can see if you compare both, if you overlap them both, you can see C is our right answer. Okay, the distance between the substrate and the transition state is less in the C graph, hence we can say C is going to be our right answer. The next question, if the product is less than substrate, then the reaction is exothermic, endothermic, thermic, thermodynamic. We know it is the exothermic. Okay, yes. Enzymes work by increasing the activation energy, lowering the activation energy, stabilizing the activation energy. None of the above. We know they mainly work by decreasing or lowering the activation energy. The next one, which of the following graph depicts the effect of a substrate concentration on enzyme activity? Okay, and that is we are having A, B, C, and D, right? So here we are having a sigmoid curve based graph, and here we are having an exponential J-shaped graph, and here we are having a hyperbolic graph, and here it is completely a straight line, right? So the graph, if you look over here, the C option is more appropriate, right? Because you can see that the graph increases, that line increases as the concentration increases, but however it becomes constant even the substrate increases than that of the enzyme concentration, right? Yes. So C is the more right option for this particular question. Next, the velocity of the reaction will not increase further even with the increase of substrate, enzyme, cofactor, coenzymes. So we know that it will not increase even with the increase in the substrate, right? So A is our right answer. Next one, match the following. Okay, and the first column we are having is transferase, hydrolase, lyase, ligase, isomerase, the different classes of enzymes and what functions they do. Geometrical interconversion. You can see that it is done by the isomerase, fifth one, right? Removal of groups other than by the hydrolysis, okay? And you can see that it is mainly done by the lyase, right? So 3 is going to be our option over here. And linking of two groups is mainly done by the ligase. Hence, we can write fourth over here. Catalysis of ether ester bonds, that is done by the hydrolase. 2 is our answer for this, and transfer of groups is mainly done by the transferases. 1 is going to be over here, right? So you can see that A, B, and C are going to be very similar in the options. Okay, the fifth one you can see, right? But however, if you check over here, yeah, you can see that yes, the third option matches, right? The third option is very much near. The fourth option is what we have written over here. So hence we can say D is the right answer for this. The next question. The cofactor that is required for the carboxypeptidase is zinc, NAD, niacin, and copper. We know it is. Mainly the zinc ion is required for this. Major of the coenzymes are ions, vitamins, prosthetic group. None of the above. Major of the coenzymes are actually vitamin in a nature, right? So we discussed around 23 questions here, and I can guess we have discussed around 103 questions if we pile up everything from all the classes. So I guess we have discussed about the enzymes quite in detail. So what you people can do, as I urge every time, that solve more and more questions, okay? And meanwhile, we will meet in the next class on the cell division and cell cycle chapter, right? Thank you.